Hello, students. I welcome you all to this class on photosynthesis in higher plants. Okay, I welcome you to this lecture. So, what is important is that you need to understand that the process of photosynthesis in higher plants is very important. So, photosynthesis, what happens in photosynthesis, where it is occurring, what is the location, where or the site, okay, or the site where it is occurring, what are the factors that are involved in photo in synthesis, okay, then what is happening if you see this word photosynthesis, it combines two words. One is photo, other one is synthesis. Okay, one is photo, other one is synthesis. So what is happening in photosynthesis is that the light energy that is obtained from the sun, okay, is utilized by the green plants, okay, is utilized by the green plants to make food, in terms of glucose molecule, and also evolve one most important thing, which is oxygen. So dear students, for studying photosynthesis you need to understand that there are several other topics which you need to understand, so such as that photosynthesis is occurring as a means of autotrophic nutrition, okay, as a means of autotrophic nutrition. And then what are the pigments that are involved? What are the pigments? What are the pigments that are involved in photosynthesis? Then, of course, we'll be looking towards the photochemical and the photochemical and the biosynthetic phase. Okay, biosynthetic phase. All right, and then we'll be seeing what is the difference between cyclic versus non-cyclic, cyclic versus non-cyclic, photophosphorylation in green plants. Then we'll be looking towards chemoisomotic hypothesis. Then we'll be looking towards the C3 cycle, okay, then C4 cycle, and of course we'll be looking toward the C2 plants where the photorespiration takes place. I will explain you all the terms which are involved, but please you need to understand that the how we'll be going through the class, what will be the flow of the class, okay, what will be the flow of the class. So it is important to understand, and of course in the end we'll be looking towards the factors that affect the photosynthesis. All right, factors that affect the photosynthesis. So what is happening is that the most important thing is, if you understand the importance of green plant, you will be able to relate that why photosynthesis is very important. Why photosynthesis is very important. Okay, why photosynthesis is very important. So if you break the word of photosynthesis, okay, if you break this word, if you break this word into photo and synthesis, it means light and something is getting synthesized. What? Food in terms of glucose. So what is happening actually is that 6 molecules of carbon dioxide along with 12 molecules of water molecule, they form glucose and release water and oxygen, utilizing the light from the sun and the pigments Okay. So these pigments are what? These pigments is none other than chlorophyll. Chlorophyll molecule is there, and it is there in the chloroplast of the green plants. All right. So what is happening is that we need to understand that how this process of photosynthesis came to the light. Who were the people that worked towards the discovery of this process of photosynthesis? So let us look at some of the people, some of the scientists which were involved in this process. So the most important one you can say is Engelmann. Engelmann was there. So what Engelmann did was that the first action spectra was discovered by him, and during photosynthesis. Okay, during photosynthesis. Okay, then Ruben and Kamen, along with the acid, used radioactive. They used radioactive oxygen, that was labeled with radioactively, and they found that the oxygen that is evolved in the photosynthesis, it comes from the water. And which plant they use? They use chlorella. They utilize the chlorella plants. So let us now peep into the core concepts and how photosynthesis is taking place and under what condition, what is the general process of photosynthesis. So as I told you, that photosynthesis is basically, it is occurring in the chloroplast, and in the chloroplast what is happening is that if you see the structure of the chloroplast, okay, it is, first of all, it is a double membrane structure. Okay, it is a double membrane structure. Okay, it is a double membrane structure, and then there are stacks. Then there are stacks over here, and these stacks are joined. Okay, these stacks are joined, and the membrane system is, this is the membrane system. This is the outer membrane. This is the inner membrane. Now this membrane system, it is concerned with the light reaction. I will explain to you what is the light reaction, what is the dark reaction. This membrane system is concerned with the light reaction. And what happens in the light reaction? There is synthesis of ATP and NADPH along with hydrogen ions are released. And this region is known as the stroma. This is the stroma. So it is associated with the dark reaction. It is associated with the dark reaction. And what happens in the dark reaction? Dark reaction utilizes the product of the light reaction to bring about the reduction of carbon dioxide, Now there are some of the pigments that are involved in photosynthesis. Okay, as I explained to you, there are some of the, let us look at the photosynthetic pigments. So the photosynthetic pigments are basically of, in the cases of higher plant, there are chlorophyll. There are chlorophyll molecule, and then there is carotenoids. So carotenoids consist of carotenes and xanthophyll. Then there is chlorophyll, maybe division hota hai. It is divided into chlorophyll A, chlorophyll B. So the peculiar thing about chlorophyll is that it is occurring in the chloroplast. This pigment is occurring in the chloroplast, and there is a differentiation between chlorophyll A and chlorophyll B. So chlorophyll A and chlorophyll B. Chlorophyll A, the color is blue-green. The color is blue-green, whereas it is yellow-green over here in case of chlorophyll B. Now the wavelength, the maximum wavelength is around 430 to 662 nanometers in case of chlorophyll A, whereas it is around 430 to 644 nanometers in case of chlorophyll B. In case of carotenoids, there are two types, as I told you. One is carotenes, other one is xanthophyll. And there is a technique. Technique is known as chromatography. Technique is known as chromatography. And in chromatography, we use chromatography to separate these pigments. To separate these pigments, chromatography technique is used. What is the detail of it? We'll talk more about it. Now, there is a few terms such as action spectra. Action spectra, there are few terms such as action spectra and absorption spectra. Okay, action spectra and absorption spectra. Then there is quantum requirement. There is quantum requirement. And then there is quantum yield. Quantum yield. So what is the action spectra? Basically, the absorption spectra, it is a graph. Absorption spectra, it is a graph. And that graph shows the amount of light of different wavelengths. Amount of light of different wavelength. Amount of light of different wavelength that are absorbed by the pigment. That are absorbed by the pigment. All right. Then in the case of action spectra, it shows the rate of a process at different wavelength of a light. So action shows the rate of a process. Rate of a process at different wavelength of light. Quantum requirement is, as you can understand from the word quantum, जैसा कि आप quantum शब्द से समझ रहे होंगे, it means quanta of light. That how many light quanta are required for the production of one molecule of oxygen? How many quanta of light? How many quanta of light is needed for the production of one molecule of oxygen, or to reduce one molecule of carbon dioxide? Quantum yield is the number of oxygen produced per quantum of light absorbed. So it is generally 1/8 or 12%. That represents the rate of the photosynthesis. And the quantum requirement is 8. Quantum requirement is 8. So dear student, let us now understand that if this is a graph, okay, and you can draw like this, okay, this is how the peaks of different pigments will be shown, okay, all right. So which is what you have to identify. Some of the questions have been asked like this. So this one is your chlorophyll B, okay. This one is your carotenoid. This one is your carotenoid. Okay, this one is chlorophyll B. I'm sorry, this one is your carotenoids. This one is your carotenoids, and this one is in the beginning. The graph is over here. This one is chlorophyll A. Now this is the absorption spectrum. Okay, okay, dear students. This one is the absorption spectrum. Now let us understand the mechanism of photosynthesis. This is some of the concepts which we'll be telling in detail. If there is some doubt, you can ask me. Whereas, let us start the process of photosynthesis. Now, the mechanism of photosynthesis, what is the mechanism? That there are two phases. One is the light reaction. One is the light reaction. Other one is the dark reaction, as I told you. Okay. So what is happening is that you need to understand that the light reaction is also the photochemical phase. इसको हम लोग photochemical phase भी कहते हैं, photochemical phase. All right. Now this photochemical phase, it occurs in the thylakoid of grana. Okay, thylakoid of grana. And जो भी events होते हैं जिसमें कि light absorption होता है, जैसे कि water का splitting हो गया, आपका water splitting हो गया, oxygen release हो गया, O2 release हो गया, and then there is production of ATP and NADPH. All right. तो ये सारी चीज होती है लाइट रिएक्शन में. अब यहाँ पे जो पिगमेंट्स होते हैं वो दो डिस्क्रीट फोटोसिंथेटिक हार्वेस्टिंग कॉम्प्लेक्सेस में, जिसे हम लोग लाइट हार्वेस्टिंग कॉम्प्लेक्सेस कहते हैं, लाइट रिएक्शन में देयर आर टू डिफरेंट discrete light harvesting complexes. Within there are two photosystem. Within there is photosystem 1 and then there is photosystem 2. All right. जो पिगमेंट सिस्टम होते हैं वो कंपोज होते हैं ऑफ क्लोरोफिल ए एंड there is this light harvesting antenna molecule that is bound to the proteins. तो क्या होता है कि इस केस में जो हमारे ये मॉलिक्यूल्स हैं, जो हमारे पिगमेंट मॉलिक्यूल्स हैं, ऑलराइट, जो हमारे पिगमेंट मॉलिक्यूल्स हैं, जब फोटोन लाइट का पड़ेगा इस पर, जब फोटोन लाइट का पड़ेगा इस पे इन द फॉर्म ऑफ एनर्जी, ओके, सो यहां से क्या होगा कि इलेक्ट्रॉन्स ट्रांसपोर्ट होंगे और ये इलेक्ट्रॉन्स ट्रांसपोर्ट होते हुए हमारे इलेक्ट्रॉन का जो प्राइमरी एक्सेप्टर होगा उसके पास पहुंच जाएंगे. ओके, और ये एक होगा थ्रू अ रिएक्शन सेंटर. दिस विल बी थ्रू अ रिएक्शन सेंटर. एंड ऑफ कोर्स दीज आर द पिगमेंट मॉलिक्यूल्स. सो लाइट रिएक्शन मैंने बताया आपको कहाँ होती है. लाइट रिएक्शन होती है इन द थैलाकॉइड ऑफ द ग्राना. इसे हम लोग फोटोकेमिकल फेज भी क्यों कहते हैं? क्योंकि एटीपी सिंथेसिस होती है, NADPH प्रोडक्शन होता है, O2 रिलीज होती है, वाटर की स्प्लिटिंग होती है. ठीक है. अभी हम लोग लाइट रिएक्शन पढ़ रहे थे. लाइट रिएक्शन में हमने दो फोटो सिस्टम्स के बारे में चर्चा करी, जो कि फोटो सिस्टम 1 है और फोटो सिस्टम 2 है. तो फोटो सिस्टम 1 जो होता है वो ऑब्जॉर्व करता है लाइट ऑफ P700. अब वी विल बी स्टडिंग अबाउट द फोटो सिस्टम. व्हाट आर द फोटो सिस्टम? तो फोटो सिस्टम जो आपका फोटो सिस्टम 1 है, then there is photosystem 2. This is the reaction center which absorbs the light at P680. 680 is the nanometer length, and this reaction center is P700. Location is, it lies on the outer surface of the thylakoids. All right, it occurs on the outer surface of the thylakoids, whereas this occurs in the inner surface of thylakoids. All right. ये दोनों में फाउंड होता है, इन द ग्राना एज वेल एज इन द स्ट्रोमा में, बट ये ओनली इन द ग्राना लैमिले में फाउंड होता है। ऑलराइट, तो रिएक्शन सेंटर इसका था P700, इसका था रिएक्शन सेंटर P600. This is, we are talking about photosystem 1 and we are talking this thing about photosystem 2. Now there are few terms. There is